Good afternoon. I'm Dr. Brian Shapiro. Thank you for joining us for the latest installments in the ATCC Excellence in Research webinar series. It's entitled ATCC's Toxicology Portfolio, Tools for Every Stage of Your Preclinical Drug Development Workstream, presented by Mr. Kevin Grady and Dr. Kevin Tayo. In this presentation, our experts discuss the characteristics and utility of various respiratory, skin, and kidney models in toxicological assays. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. Any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our speakers.
Thank you, Brian, and welcome to those in attendance. Kevin and I would like to take this opportunity to introduce you to some of ATCC's portfolio for toxicology. We will focus first on what is ATCC's mission and the future direction in the areas of toxicology. Then we'll delve a bit deeper into ATCC's airway, dermal, and kidney models for toxicology. But first, a brief intro into ATCC. ATCC is a not-for-profit organization and the world's largest repository of biological material and information supplying authenticated microbes, viruses, and cells that have become recognized as gold standards in research. That may be how most of you think of ATCC, but we are much more than that. We have a talented R&D team focused on developing new and innovative products employing today's novel technologies. This dedication to innovation allows ATCC to provide new gene-edited models with unique reporter systems, 3D cell models, and in vitro models with more in vivo biological relevance, such as those we'll be discussing today. In the past, ATCC built its collection by cells and microbes being deposited by research scientists. That still happens today, but ATCC's dedicated research and development teams are focused on developing and producing new products to meet the growing and ever-changing needs of the community. But we're also focused on investing in new technologies so that ATCC's future products and services will still remain the definitive standards for biological research in the future. In vitro cell models have grown in complexity and functionality. It all started with continuous cell lines isolated from tumors or discovered through spontaneous or induced mutation. Next came transfected or transduced cell lines that we genetically manipulated along with the development of primary cells, which are isolated from primary tissues. This did require the development of specialized isolation procedures and media. The next generation of cell models included gene-edited cell lines using such technologies as CRISPR, along with the development of immortalized primary cells, which combine primary cell physiology with the growth properties of a continuous cell line. And the current stage of model development is focused on 3D, in either organoid structure or 3D models involving single to multiple cell types. The choice of which model type to use can be challenging. Every cell model has its pluses and minuses. This table presents various attributes for each type, and depending on where you are in your research workflow, a certain cell model may be better than another. Starting on the left, we have continuous cell lines. These are routinely isolated from a tumor, so they have great proliferative capacity, but are generally very robust and easy to culture. However, they have poor genetic stability, which results in low biological relevance and low predictability in tox studies. Next are primary cells, which are isolated directly from normal primary tissue. These cells are diploid and exhibit great genetic stability. However, since they are primary cells, they do have a limited lifespan in culture, often only 10 to 15 population doublings or 6 to 10 passages. So they offer limited experimental reproducibility, as you can only get a finite number of cells out of one specific donor organ. But primary cells do offer the highest level of biological relevance by closely mimicking the in vivo state and conferring the greatest toxicological predictability. The last model on the table is HTERT immortalized primary cells. These are genetically modified primary cells such that they retain the near normal primary functionality while exhibiting the growth and proliferative capacity of a continuous cell. Also, each line is derived from a single clone, so there's limited variability introduced from lot to lot. These cells offer the benefit of both worlds, combining the best of continuous cells with the best of primary cells. As I stated, every model has its pluses and minuses. If you are performing a large screen and need lots of cells, continuous cells may be the best. As your targets narrow, you may want to screen, but with a more biologically relevant and predictable model, so HTERT cells would be your model of choice. Now that you're down to a few likely candidates, you want highest predictability, so you move to a primary cell model. Now that you're familiar with all the possible cell models, let's discuss more about the two most predictive models. First, we'll focus on primary cells. ATCC supplies a complete portfolio of primary cell types isolated from human solid tissue and blood. As ATCC is well known for its cell authentication and characterization, all primary cells are characterized by testing for both positive and negative markers to assure high isolation purity. Primary cells are also biofunctionally characterized and their functionality is confirmed. ATCC primary cells are supplied at P0 for blood cells and for P2 from cells isolated from solid tissue. Our primary cells are guaranteed for greater than 10 PDLs. Now some background on HTERT immortalized primary cells. Normal somatic cells have telomeres at the ends of their chromosomes. As the cells grow and divide, the telomeres are lost and the cells eventually senesce. That is why primary cells have a finite lifespan. In cancer-derived cells, telomerase is active and blocks the telomere erosion, so the cells never enter senescence. For HTERT immortalization, the catalytic subunit of telomerase is stably transfected into a primary cell. Then, through antibiotic selection and clonal isolation, stable clones are generated. ATCC then screens the clones, looking for the one with the best primary cell functionality and the least chromosomal instability. HTERT primary cells are fully characterized, just as the parental primary cell, except HTERT cells are guaranteed to express primary cell functionality at longevity, which is considered to be greater than 35 PDLs. ATCC has models including continuous cells, HTERT, immortalized primary cells, and primary cells to support your research from start to finish, from basic research through to discovery and development and on to product testing. ATCC's portfolio is fully authenticated and characterized. And now I'll ask Dr. Kevin Tayo to discuss his work with ATCC airway cells.
Thank you, Kevin, and good day, everyone. My name is Kevin Tayo. I'm a scientist at ATCC. I wanted to continue Kevin's discussion on ATC's products with toxicology and talk about our work on fabricating and assessing the functionality of bronchial tracheal airway models. Here at ATCC, we have a variety of primary cells for use in research for the respiratory tract, such as bronchial tracheal, small airway, and lobar epithelial cell lines, as well as lung smooth muscle cell lines and lung fibroblasts. We also have in our product catalog disease primary cells for asthma, COPD, cystic fibrosis, and pulmonary fibrosis. In addition, we also offer HTERT immortalized bronchial tracheal and small airway epithelial cell lines, as well as fibroblasts. For these studies, we fabricated airway models comprised of either primary bronchial epithelial or HTERT immortalized bronchial epithelial cell lines, as well as fabricate models with or without the incorporation of primary or HTERT lung fibroblasts. Here's a schematic providing an overview of our airway model fabrication process. All of our models are grown in 24-well plates with transwell inserts possessing 0.4 micron PET membranes. First, 3 mg per ml collagen solution is used to coat the apical side of the inserts prior to cell seeding, followed by fibroblast addition at 20,000 cells per well. The following day, we then add the undifferentiated bronchial epithelial cells to the apical side as well at 100,000 cells per well, and allow the cells to reach full confluency, which typically takes about 48 to 72 hours. Once the cells are confluent, we completely remove the apical side media and expose the epithelial cells to an air-liquid interface, or ALI, culturing condition. We keep the cells under air-liquid interface culturing conditions for about 4 to 5 weeks to induce primary cell differentiation, while the basal side media is replaced periodically about 3 times a week. Once this is accomplished, the models are checked and are then considered ready for testing. Following the fabrication process, we first want to assess the morphology of the cells to visually confirm the presence of basal, goblet, and xylated cells in our mature airway models. Both Alcian blue and H&E staining are performed on histological sections of airway models. Images of the sections are taken, with Alcian blue allowing for the distinction of goblet cells, whereas H&E staining allows for the user to visually distinguish xylated cells from the other cell types. Here are images taken from scientific literature demonstrating that these airway models also have a pseudo-stratified structure. Here are some representative images of stained sections taken from some of our in-house generated airway models. Specifically, we're looking at images from models comprised of only primary epithelial cells, models comprising of both primary epithelial and HTERT fibroblasts, as well as models comprising of HTERT bronchial epithelial and HTERT lung fibroblasts. The top 3 images are from sections stained with Alcian blue, with all 3 models showing the presence of goblet cells, shown here as the blue stained cells. The bottom images show a similar story, with all 3 models showing the presence of xylated cells circled here for your convenience. In tandem with imaging work, we began to assess the functional properties of our airway models by testing their tight junction during air-liquid interface culturing. To do this, we conducted weekly measurements of each model's trans-epithelial, trans-endothelial electrical resistance, or TEER, using an ohm meter. Increased TEER values correspond with increased tight junctions. Additionally, we also assessed the barrier function of our airway models using fluorescently labeled dextran with a 40 kilodalton molecular weight. We first introduced the compound on the apical side of the transwell insert and then examined the changes in basal concentrations of dextran at specific time points. Thus, we're able to assess the differences between each airway model's transwell kinetics. Here is a slide to provide some more additional background information on TEER measurements in bronchial airway models. These images were taken from scientific literature and demonstrate that TEER values from primary bronchial epithelial airway models can vary. Here are more images from scientific literature with previous work showing that there are TEER differences between models of primary bronchial epithelial cells versus HTERT bronchial epithelial airway models. Here are the results from our weekly TEER measurements taken from our airway models. I know this is a busy slide, so let's break it down. I first wanted to mention that we culture these models using a medium mixture comprised of 80% airway epithelial cell basal medium and 20% fibroblast basal medium, with each medium supplemented with their respective growth kits. As you can see on the x-axis, we tested models with 2 different HTERT epithelial cell lines. HBEC3-KT and Newly-1, as well as HTERT models that incorporate either primary or HTERT lung fibroblasts. We have on the right side of the x-axis models made with primary epithelial cells, with or without primary or HTERT lung fibroblasts. For each model, there are four bars that correspond to weekly TEER measurements, with each week showing how long the models were under air-liquid interface culturing conditions. First, we noticed that all of our models required either 4 or 5 weeks of air-liquid interface culturing to provide maximum TEER values, and that Airway models comprised of primary bronchial epithelial cells demonstrated a marked increase in absolute TEER values, or ohms times centimeters squared, relative to models of HTERT cell lines. In addition, we noticed that the incorporation of fibroblasts did not measurably affect the absolute TEER values, nor the overall trend in tight junction function development. In this slide, models were cultured in media mixtures of 50% complete bronchial and 50% complete fibroblast media. So the only difference here is the medias used in culturing these models were a different ratio. And yet, we see a similar story where Airway models comprised of primary epithelial cell lines show a greater absolute TEER value relative to HTERT models. And again, that fibroblast incorporation into our Airway models does not alter or hinder tight junction function development. On this busy slide, we show results from our dextran kinetic study 5 weeks following initial air-liquid interface culturing. At 2, 4, and 6 hours following dextran administration on the apical side, we collected 10 microliters of basal media to assess changes in dextran concentration relative to models of primary epithelial cells, shown here as the white bar. What this graph is showing is that after following 6 hours after initial dextran administration, models comprised of HTERT epithelial cells show a marked increase in dextran concentrations in the basal side, indicating that the tight junctions in these models are less developed compared to our models comprised of primary bronchial epithelial cell lines. This data corresponds with our TEER values, with the model's relative fluorescent values are inversely proportional to their model's TEER measurement values. Following these functional studies, we began exploring how well these models will react to exposure to toxins. Right now, we're in the process of conducting toxicology screening assays using mature Airway models. However, I can share some of our preliminary work using undifferentiated primary cells grown in transoral inserts. For these initial preliminary toxicology tests, we assessed the response of early Airway models to cadmium chloride exposure. Cadmium chloride was chosen due to its chemical stability and low vapor pressure. Here, we exposed cells to cadmium chloride concentrations ranging from 43 micromolars to 1.4 millimolars. These concentrations were based on previous scientific literature. In these studies, selected cadmium chloride concentrations were administered to the apical side of our Airway models, followed by the immediate placement of these plates in a biospa citation system shown here for real-time measurements. Measurements and viability changes were conducted using commercially available assay kits. In both assays, increased signaling corresponds to increased cell death. For our first test, we wanted to assess changes in cell viability using Promega CellTox green cytotoxicity assay kit. Following cadmium chloride administration, a fluorescent dye is added to the apical side of the transoral insert. If a dye binds to DNA, the fluorescent properties are enhanced. Increased fluorescence is associated with decreased cell membrane integrity and ultimately cell viability. These images here were taken from plates immediately following cadmium chloride administration and show, with the exception of lysis controls, there is little to no fluorescence or cell death present at 0 hours. However, as time progresses, there is a dose-dependent response to cadmium chloride exposure, with wells exposed to the highest cadmium chloride concentration, shown here in the top left, demonstrating increased cell death following a 24-hour incubation time. These trends are quantified on the next slide. Here, we graphically represent the data from the previous slide to show a dose-dependent response to cadmium chloride exposure. Here the x-axis shows that the time points where measurements were taken, with each value corresponding to a total fluorescent response that has been corrected using the untreated blank controls. We see a dose-dependent response to cadmium chloride exposure exemplified in sample 1, which has the highest concentration of cadmium chloride. During a 24-hour period, the fluorescent intensity from sample 1 steadily rises, reaching a similar intensity to our licensed controls, which demonstrate the maximum signaling in cell death. The other samples also follow this trend, but to a lesser intensity, again showing a dose-dependent response to cadmium chloride exposure. We conduct another assay using Promega's LDH Glow cytotoxicity assay. Here samples, about 20 microliters worth, from the basal side are taken at 24 hours after cadmium chloride administration. Samples are diluted in a storage buffer to a final volume of 1 ml and then mixed with an LDH detection reagent, causing an enzyme-coupled reaction generating luminescence proportional to the LDH concentration of the sample. This graph shows similar results to the previous assay, with a dose-dependent response and viability changes from cadmium chloride exposure, with increased cadmium chloride concentrations corresponding to increased cell death at 24 hours following toxin administration. Finally, I wanted to share the results from our final Airway model toxicity study. Here, we wanted to assess H-TERT lung fibroblasts response to chlorohexidine exposure. We showed that the primary lung fibroblasts demonstrate a dose-dependent response to chlorohexidine, with increased chlorohexidine concentrations corresponding to increased cell death relative to untreated controls. We also show that our H-TERT immortalized lung fibroblasts demonstrate a dose-dependent cytotoxic response as well, and this profile is similar to our primary cells. In conclusion, Airway models comprised of bronchial, epithelial, and fibroblasts from H-TERT and primary cell types were successfully constructed. Histological imaging demonstrated that long-term air-liquid interface culturing resulted in the maturation of models via the presence of differentiated bronchial epithelial cells. TEER measurements showed that differences exist between models comprised of primary and H-TERT epithelial cells. Fibroblast incorporation did not affect TEER readings, and altering media ratios made no difference in changing TEER values. Dextran transportation results were inversely proportional to TEER measurements. And finally, preliminary toxicology testing showed that the initial Airway models in the H-TERT fibroblasts provide a dose-dependent response to cadmium chloride and chlorohexidine exposure, respectively. Studies are currently underway to assess toxic response for mature Airway models. Thank you all for your time. I'll let Kevin Grady continue with this presentation.
Thank you, Kevin. Now I'm going to talk a little about ATCC's dermal models. ATCC offers a number of different types of dermal cells. We offer keratinocytes, melanocytes, and dermal fibroblasts in both primary cell and H-TERT immortalized cell format. Primary and H-TERT immortalized cells can be used to generate 3D organotypic models in vitro. To do that, a matrix of Eagle's medium, rat tail collagen, and dermal fibroblasts was cast onto transwell inserts in deep well plates and cultured for 7 days. Dermal keratinocytes were then seated on top of the matrix, and epidermalizing media was added to the apical and basal chambers of the transwell. After 2 days, the media in the apical chamber was removed to create an air-liquid interface. The cells were allowed to differentiate for 11 to 21 days post-airlift, at which time they were fixed, cross-stained, and H&E stained, or stained with specific markers using primary antibodies directed against keratin 14 and filigrin. As shown by H&E staining, primary keratinocytes developed into characteristic stratified epidermis architecture composed of proliferating basal and differentiated suprabasal keratinocytes, as well as an evident superficial stratium corneum after 11 days in air-liquid interface culture. H-TERT immortalized and primary keratinocytes differentiate into similar stratified epidermis architecture. To verify the full differentiation of the cells, frozen cross-sections were stained with antibodies directed against filigrin, which stains for differentiated keratinocytes, and KRT14, which stains for epidermal cells. Filigrin in both primary keratinocytes and H-chart immortalized keratinocyte models displayed increased positivity in the cells that are closer to the surface and nearing terminal differentiation. Both primary keratinocytes, H-chart immortalized cells displayed KRT14 reactivity throughout the basal and suprabasal epidermal structure. This data demonstrates that the immortalized keratinocytes retain differentiation capability after an extended in vitro culture period beyond the capacity of normal primary keratinocytes. Thus, H-chart keratinocyte cells have significant value for a wide variety of investigations of human epithelial biology in a tissue physiologically relevant context. The organotypic model generated in the previous slide can be used in toxicological studies. Triton X100 is considered to be a strong skin irritant. In this study, Triton X100 was added to both primary and H-chart 3D organotypic models. As you can see in the graph, viability decreased as the time of exposure increased. Both models exhibited similar levels of toxicity, thus showing that an H-chart model offers equivalent performance to a primary cell-generated model. Another important dermal cell type in our portfolio are H-chart immortalized melanocytes. We currently offer H-chart immortalized adult melanocytes and neonatal H-chart melanocytes. These cells are fully characterized, exhibiting the positive expression of TEER1, a key enzyme in melanogenesis. We also verified that immortalized melanocytes exhibited melanin expression at over 50 PDLs, thus confirming that H-chart immortalized primary cells maintain primary cell functionality. With the addition of melanocytes to the portfolio, we could now establish a 3D organotypic model comprised of H-chart dermal fibroblasts, keratinocytes, and melanocytes. This model was constructed using our neonatal melanocytes, and the model was constructed as previously described. Two models were created. The first model had all three cell types: H-chart immortalized fibroblasts, keratinocytes, and melanocytes. The other model only had fibroblasts and keratinocytes. This was done so that one could clearly see that melanogenesis had occurred due to the presence of melanin in condition 1, as shown by the yellow arrows pointing out the deposits of melanin. ATCC also offers gingival cell types: primary gingival keratinocytes and fibroblasts, and H-chart immortalized primary gingival fibroblasts. In this study, we again show that H-chart immortalized cells exhibit similar toxicities as their primary cell counterparts. Here we exposed primary gingival fibroblasts and H-chart gingival fibroblasts to a low dose of chlorhexidine at 1 micromolar or a high dose at 10 micromolar and measured viability over 3 hours. The observed toxicities were similar in both cell types at all conditions. Now we will discuss our products for nephrotoxicology. ATCC supplies primary H-chart immortalized RPTEC cells, which are renal proximal tubule epithelial cells. Both cell types exhibit functional characteristics such as positive expression for ECAT-herin, an epithelial cell marker, and CD13, an RPTEC marker. The cells exhibit stabilized TEER, which measures electrical resistance across a monolayer of cells. This confirms integrity and permeability of a monolayer. Additionally, both cell types exhibit dome formation, which is indicative of a transport or secretory function in vivo and in vitro. Organic anion, and cation transport function is lost in primary RPTEC cells in culture. This transport function is vital in kidney metabolism. So ATCC created enhanced models for kidney research. We took our HTERT immortalized RPTEC cells and stably transfected them to express either OAT1, OCT2, or OAT3 transporters. The expression of these transporters were confirmed at the genomic, proteomic, and biofunctional level. To confirm functionality of the models, we performed an uptake assay using fluorescent membrane impermeable dyes, 6-carboxyfluorescein for OAT transporters, and ethylamine iodide for OCT2. Parental and transporter cells were tested with increasing concentrations of either dye. The green line represented the parental HTERT RPTEC, and the purple line is the transporter model. In all cases, the parental had little to no transport activity, while the models which stably expressed the transporter proteins had significantly more transport shown and in a dose-response manner. Next, we wanted to show that transport was specific, so a similar experiment was set up, but this time the uptake substrate concentration was constant, but we added increasing concentration of a known anion transporter inhibitor, novobiocin, to the OAT cultures, and a known cation transport inhibitor, cetamidine, to the OCT cultures. As you can see, in all cases, a dose-dependent inhibition of uptake was observed. So to conclude our presentation, we hope you can now appreciate ATCC as your complete solution supplier for toxicology, as we supply fully authenticated and characterized models for all stages of your workflow, from continuous cell models to more enhanced models with greater biological relevance and predictability, such as HTERT, immortalized primary cells, and primary cells. We also presented data that supports either using primary cells or HTERT cells for most applications requiring high biological relevancy. On our website at www.atcc.org, you can find resources to guide you through the use of ATCC products in your lab to improve your productivity and the reliability of your research.
All right, well, thank you, Kevin. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. This session will be archived on the ATCC website at www.atcc.org. Also, our lead scientist in ATCC's toxicology group in research and development, Sujoy Lahiri, has joined us. So if we're having any problems answering the questions, he may jump in. So let's go ahead and jump into the first question. This is definitely firmly in Kevin Tayo's arena. Kevin, why were fibroblasts added to the Airway model, presumably the 3D one?
Yeah, that's a great question. We wanted to provide a more physiologically relevant model, and the idea is that they would actually, in the future, in future toxicology studies, they would provide maybe a different response to toxins relative to 3D models with just epithelial cells alone.
Okay. So then I guess that's, specifically, but just generally, is it possible then to add additional cell types into the Airway models?
Yes, of course. One of the cool things about this way to fabricate these models is that you can add additional cell types, not just fibroblasts or epithelial cells, but other things such as endothelial cells, even macrophages as well, just to kind of tailor suit the responses that you want to look at in regards to the multiple different, excuse me one sec. Yeah, the multiple different responses to different types of variables that you want to look into. So again, it could just be toxicology response, or if you want to look into immune response to, let's say, introduction of bacteria or viruses.
Gotcha, gotcha. And, you know, here's another question around primary lung cells. Did you notice in your experiments any donor variability that was introduced?
Yes, that's a good question. So donor variability will play a role in the responses that you see from when using primary cells. Now, if you start looking at overall trends, yes, the trends are going to be the same. If you look at, let's say, toxicology, testing where you're assessing toxin response, yes, higher concentrations typically results in increased response or cell death. But, the overall values are going, or the specific values are going to be different. And again, this is because you're going to see donor variation. Donors will have, let's say, different genders, ethnicities, as well as age, and that's going to really affect the absolute values of responses. But again, the trends will overall be the same.
Okay, no, that sounds good. That sounds good. So this next question isn't really related to the Airway models, and basically it's just asking, do we have any neuronal models that are H-TEER immortalized? And just long story short, we do. We have some Schwann cells that ATCC accessions through NTAP, which is the Neurofibromatosis Therapeutic Acceleration Program. So what we have is H-TEER immortalized wild-type NF1 and neurofibroma-derived cells that harbor NF1 mutations. So these are all highly genotypically characterized, and with generous support from NTAP, the purchasers from these cells are eligible to receive some reimbursement from NTAP. So, and that's essentially the extent of the neuronal models that we have. So I guess I'd like to switch gears now and maybe cover some questions that are around the RP-TEC cells. So I guess let's first start out with this. One is around special media to culture the RP-TEC cells. Is there any special media that you need?
Oh, I guess this would be for me, right? So for the transporter cells, yeah. Okay,
Right.
So so the media, from my understanding, should be the same as the parental cells, but we do suggest that you add, I think it should be about 0.3 micrograms per ml pyromycin solution for just provide like a suction pressure.
Okay, good, yeah. And that media, its SKU is ATCC ACS 4007 if you are interested. And then this next question is about passaging. So how long can the RP-TEC SLC transporter cells be passaged?
Well, I do know we verified it where it could be done for at least 10 passages. Now, granted, these cells, since they're immortalized, in theory, they can be passage indefinitely.
Okay, but tested at ATCC for up to 10 passages then?
Yes.
All right, good. Now, so I kind of like this question a lot. How many copies of the transporter gene insertion are actually in the cells? And how do we verify that?
Yeah, that is a good question. So we did verify the assertion copies by PCR, and it shows there's, I think, 5 extra copies of the OAT1 gene insertion. And there's 3 copies of OCT2. So that, and again, we verified this through the PCR. So those, it's 5 and 3 respectively for OAT and OCT2.
All right, good. I like it. Now let's see. Okay. Have we ever tried expressing both OAT1 and OCT2 in the same line? And if I can actually jump on that question, we've tried it and it just didn't work for whatever reason. So we have, but nothing to report from that. Okay.
That's right.
Let's see. Yeah. I guess let me go ahead and jump to a different question that's just generally about H-turn immortalized cells. So how long can an H-turn immortalized cell be in culture for?
Yeah, that's a great question. So again, our H-turn cells, I know that we test them out for at least 35 PDLs, but we've routinely will culture them way beyond that, something like 250 population doublings, and that will, and we've shown that they will show no loss of like change or performance, so they'll still stay the same. So you can keep these things for quite some time.
Okay. Good. Now here's I have a specific question. Can all cells respond to H-turn telomere modification to become immortalized? I guess a better way of putting this would be, can you just introduce the catalytic subunit of telomerase into any cell and expect that it'll be immortalized, or does more need to be done in some cases?
Mm-hmm. Well, I'm not an expert at this particular thing, but I do know that we do prefer to use H-turn alone to immortalize cells. But I do know that there are some cases where it's not enough. So for example, certain cell types, it does require additional modification. So I think like keratinocytes, they require expression of like a modified H-turn as well as a CDK4. So it's not a universal thing, but we do prefer that.
Gotcha. All right. Let's see. Now I think that this question was answered in the presentation, but Kevin, do H-turn dermal melanocytes lose melanin expression?
No. So the actual melanin expression can be observed and quantified, during the subculturing, when we like pelt cells in their suspension. So prolonged culturing of immortalized primary cells may reduce melanin expression, but we've observed a decrease of melanin if the cells are allowed to become fully confluent. But we don't really see a complete loss of melanin with these H-turn immortalized adult melanocytes.
All right, good. Now, Kevin, is the Airway air-liquid interface protocol, the ALI protocol, available online?
Yes, we actually do have an application data that is available on our website. It provides a detailed protocol on the air-liquid interface of these 3D cultures using these transwell plates. So, we're specifically, this protocol shows the differentiation of bronchial, tracheal, epithelial airway models. Mm-hmm.
Right, and there's actually another application note out that describes the toxicological studies that you performed as well, correct?
Yes, that is correct.
To allow you a little shameless self-promotion there.
Okay. Yeah.
So, and I wanted to sort of use that to transition into this next question, which has just come in, about the concentration of cadmium chloride. The asker asks, why such a high concentration of cadmium chloride? I mean, is this physiologically relevant? And what made you choose it?
We wanted to make sure that we have a wide enough range to where we would ensure complete cell death on the highest level, as well as exceed, the IC50 that is reported in literature, but at the same time have also a low enough concentration that it wouldn't provide or produce noticeable effects.
Okay, good. Good answer. Good answer. Now, this is just a general question, I guess. Can HTERT cells be used on flasks without, say, a lysine coating? I kind of feel like there's sort of two answers to this. The first answer is yeah. The second answer is, though, well, it depends if that specific cell type requires a lysine coating, then you would need that. But I don't believe any of them specifically just to grow them to propagate need a lysine coating.
Yeah, typically, the thing about HTERT cells is that they still have, they're very similar to primary cells, except they just are immortalized. So the media itself typically is going to be the same as your unmodified cells.
Right. And so basically you would use mostly the same conditions to just get them to attach to the plate, right?
Mm-hmm. Correct.
All right. Let me see. All right. So you know that I don't know is a perfectly good answer to a scientific question. And this next question, I'm not exactly sure too much about it. I don't know very much about this subject, but are there any models available to study skin absorption properties as a substitute for OECD 428 guidelines?
Oh, that is a good question. I'm not sure about that off the top of my head.
I can take that actually.
Okay.
So, yeah, the OECD guideline is basically internationally recommended guidelines for different toxicity. That could be ocular toxicity, dermal toxicity, and so on and so forth. So it's basically the industrial guideline. So is the question directed that whether the model is available at ATCC or the model is available elsewhere in the world?
Let's go with, is the model available with ATCC?
Right. So with respect to dermal modeling, currently, as we are talking about it, we are actually working on dermal models. And so we have established dermal models already using HTERT dermal cells, as well as a combination of HTERT and primary cells. So we have used, for example, primary melanocytes along with HTERT fibroblast and HTERT keratinocytes like the CAR-CT cells. We have done that previously, and we have done some tox assays, and those models could be used, and we have some application notes in our website, and those could be used for OECD compound testing. However, currently we are working on primary cells derived from human skin, and we are trying to build the model that could be used for similar tox assays. And I guess that within next couple of months, we will reach a conclusion, and we will be releasing application notes by this year.
All right, good. But glad you joined us, Sujoy. Appreciate you being able to take that. All right. So I guess this next question is just generally speaking, how well do in vitro assays using the RP-Tech cells capture in vivo renal toxicity? And I guess there's two ways to go at this. Just RP-Tech cells in general, then H-tert immortalized RP-Tech cells, and then the RP-Tech cells that overexpress the SLC proteins. And anyone can take this. I guess I'll go ahead and jump on it then. So, yeah, I mean, primary RP-Tech cells do a pretty good job of, capturing in vivo renal toxicity, except, pretty quickly, once you take a cell, an RP-Tech cell out of the body, it stops expressing, the cellular carrier proteins. So that's one major aspect of renal physiology that's immediately gone. Okay. Now, H-tert immortalizing the primary cells isn't going to bring that back necessarily. So you're still going to be kind of missing that one aspect. And so, using the cells that have the expression of OAT1, OX2, and OAT3, that's been put back into them, that's going to be a much more, going to do a much better job of capturing in vivo renal toxicity, right? So you're actually going to be able to see those transporters transporting toxic compounds into the RP-Tech cells, and presumably that's going to increase the toxicity. All right, and then we have one last question that's come in, and this is kind of possibly from left field. I'm not sure if anyone actually has a good answer for it, but do we foresee clinical usage of oligosaccharides for health applications such as anti-cancer treatments? Again, I don't know is a perfectly good answer.
Yeah, I'm not sure. I'm not an expert on that.
We can think of possible collaboration if someone is trying to find out a model where in a cancer model they want to use oligosaccharides for treatment or oligosaccharides-based compounds for treatment. We can possibly figure out a collaboration, but I don't think that we have explored that path yet.
Okay. No, that's good. That sounds good. All right. Well, if we don't have any more questions coming in, we can conclude our Q&A session. So thank you, Kevin, for the excellent presentation. Thanks for Sujoy for jumping in and helping out with the Q&A session. And we hope to see you for more webinars that are coming up. So next week, Kevin will present Enable Renal ADME and Pharmacokinetic Studies using solute carrier transporter cells, so we can learn more about the renal transporter cells, the RP-Tech cells that express OAT1, OX2, and OAT3. We also have coming up 3D models for absorption in cosmetic toxicity studies, primary and immortalized epidermal cells, which we previewed for you in this webinar. And then we hope to see you at Society for Toxicology and Tox Expo for our exhibitor-hosted session. We'll be presenting our talk on primary and H-tiered immortalized cells, physiologically relevant models for toxicological assays, located at the Music City Center, room 101D, on March 21st, 2023, between 10:30 and 11:30. And then finally, if you want to learn more about our toxicology product, you can visit www.atcc.org/talk. So thanks again, everyone, for attending, and have a great day.